Hello, good afternoon, and welcome to everyone attending the third session of the NPRC's 60th Anniversary Scientific Seminar Series, dedicated in honor and memory of Dr. Andrew Lackner, former director of Tulane National Primate Research Center. Today's session focuses on neurodegenerative diseases, what we have learned from non-human primates, about neurodegeneration, why this is so important, and what we need to know about this growing field today. We have an incredible lineup of three speakers. We're really pleased to have these speakers today and share the cutting edge work that they're doing. And this sort of gives an idea of where we are in the field and what the enormous possibilities are in the future, and how important non-human primate research is. Non-human primates are closest to man, and this is why we use them in our studies. Their brains are very similar to ours, and some of the studies, translational studies that are done in other species, don't always translate, and things that look promising in other species may not pan out when they move into clinical trials. So this is one of the reasons why studies in non-human primates are so important. And you will learn from the talks that are coming up more detail about why this is so important and what these correlations are. Before we start, I'd like to encourage you to submit questions for our presenters so they can have so we can all have a thoughtful discussion at the end of the three talks. And the questions you can submit at any time through the Q&A function, and will be answered by our presenters in a panel discussions after the three talks, and I will be moderating and presenting the questions to the speakers. With that, our first presentation is from Dr. Jeff Cordova, founding director of the Arizona State University Banner Neurodegenerative Disease Research Center, and the Charlene and Jay Oren Edson Distinguished Director of the Biodesign Institute of Arizona State University. And he will be presenting his talk, Non-Human Primate Models for Neurodegenerative Diseases.
Hello, my name is Jeff Cordova, and it's my pleasure to talk today about non-human primate models of neurodegenerative diseases. I've been using monkey models for many, many years, and I can tell you I'm committed to their use and to the importance of their use, especially in the area of neurodegenerative diseases. So, what's the advantage of using non-human primate models? First of all, they have large gyroencephalic brains that's organized more like humans, far more than lower species. In fact, mice and rats really don't have a prefrontal cortex, for example, while monkeys have a very complex prefrontal cortex. So, also monkeys can perform complex motor and cognitive tasks. They have similar endocrine function to humans, regular cycling, and similar immune systems to humans, and quite distinct from rodents. So we can design non-human primates with the endpoints that are similar to the scales and tests used in clinical trials. So what happens when you rigorously use non-human primates to do things like find out the organization of the primate basal ganglia? Well, what can happen is what's been shown, we'll show here, is what Malin DeLong demonstrated a number of years ago following his landmark non-human primate studies, ultimately resulting in the use of deep brain stimulation for the treatment of Parkinson's disease. So here's a person off medication preoperatively for prior to deep brain stimulation. You can see this person would fall if this nurse didn't catch him. And you can see the festinating gait, and you can see this person is completely dependent and totally disabled. But when you put deep brain stimulators in the subthalamic nucleus, look what happens. It's a miracle. And this person is completely normal. And we know which patient population this will work for. And this is totally dependent upon the classic work of Malin DeLong in non-human primates. So what models are available? I'm going to talk to you about a few models for movement disorders, then we'll close with models for Alzheimer's disease. Well, one model that many people don't use is aged monkeys. Now, most of these diseases, like Parkinson's and Alzheimer's, are age-related neurodegenerative disorders. So why should we study normal aging and using aged monkeys as our model? First of all, aging is the most prominent risk factor for the development of neurodegenerative diseases, including AD and PD. They develop Parkinsonian signs and cognitive decline, which is a prominent feature of normal aging in some subjects. There's also preclinical loss of non-extracellular dopamine in normal aging. Now, I can tell you that I'm one of a few groups that use aged monkeys as a model for degenerative diseases. So is there a reason that this is really important? And let me just show you some data from an older study they performed with Tim Collier that shows that aged monkeys are fundamentally different than younger monkeys. So what we did was gave MPTP up the carotid artery unilaterally in both young, middle, and aged non-human primates. And our goal here, if you look at dopamine content, is to drive dopamine levels to virtually zero on both sides. Now, HVA dopamine ratios is a measure of compensation, meaning when you make a lesion, other neurons in the general area or projecting to general area will work harder to compensate for the loss of their neighbors. And you can see very high HVA dopamine ratios in young monkeys, non-statistically significant difference between young and middle-aged monkeys. And I can tell you that, look at this error bar here. So what's happened, this means is that some monkeys are still compensating, but others are decompensating. But look what happens in an aged animal. You can see that none of the animals will compensate. So that may be the reason why young people who get Parkinson's disease have a much more benign course, and aged humans have a much more, when they get Parkinson's disease, have a much more malignant course. And so we could use young and aged monkeys to model these two aspects, heterogeneous aspects of Parkinson's disease. But what about the pathology? So we all know that Parkinson's disease is a synucleinopathy, and we did studies in aged monkeys to demonstrate that a 7-year-old monkey, which is about a 21-year-old human, 15-year-old monkey, which is about a 45-year-old human, a 29-year-old monkey, which is about almost a 90-year-old human. And you can see and when they're young, all you can see is some fibers in the substantia nigra, and then you can see an increase in the number of synuclein-positive pericardia. And then you can see there's an increase in both the density, number, and intensity of staining for alpha-synuclein in the most aged animals. And you can see here the quantification demonstrating that fact, that you have young monkeys and you have middle-aged monkeys and aged monkeys, and you can see independently each individual case. If you look at the optical density of neurons in the nigra versus chronological age, there's a very high statistical significance and correlation between the expression of alpha-synuclein and chronological age. And I can tell you that all this synuclein here in aged animals, like aged humans, is monomeric, and it's the beginning, the prelude of the pathology that's seen in Parkinson's disease. Now, another model that we use, I'm just going to briefly go over this, and this is MPTP, and you can see a monkey treated with MPTP. If blue here means absence of dopamine, gold here is the dopamine that was protected by lenti-GDNF. But you can see here that the model of MPTP is good, not in terms of studying pathogenesis, but in terms of making comprehensive lesions where you're going to have behavioral deficits. And then you can study certain neuroprotective or restorative therapies using MPTP. I'm not going to talk more about MPTP today, but the other thing I want to talk about, though, is the pathology of alpha-synuclein and how it relates to non-human primates. And you can see here in human brain that if you have alpha-synuclein on a cell in the substantia nigra, you have decreased optical density of tyrosine hydroxylase, decreased optical density of Neur1, because one of the things that alpha-synuclein does in the brain is reduce the phenotype of dopaminergic markers. And that's the same thing that we see in monkeys that receive preformed fibrils, and I'll show you that data in just a bit. So the pathology in alpha-synuclein is the pathology in Parkinson's disease is a synucleinopathy, and this was really led initially by Heiko Brock, who demonstrated that synuclein progresses in the brain from the gut, also the olfactory bulb, and then in a prion-like manner spreads throughout the brain. We demonstrated the same phenomenon. Patrick Brundon and I independently demonstrated this in transplants of fetal neurons in patients with Parkinson's disease. But the main person who blew the doors off this hypothesis is John Trojanowski and Virginia Lee. And quite sadly, we lost John. What a giant in the field, figuratively and literally, and his partner, Virginia. And we send our condolences to them, to Virginia, about John's passing. But they demonstrated with Kelvin Luck that when you put preformed fibrils into the striatum of both transgenic and non-transgenic animals, you get this spreading of pathology throughout the brain. And these papers are classic and quite detailed, and I don't have time to go into all the details here, but I remember seeing this data. And I call up John, I go, I want to do this in monkeys. Can I come and see the original material? And I will tell you, it was just fantastic. John and Virginia and I had a great day, and we formed a collaboration to look at whether this spreading also occurs in non-human primates. And this work was being done by my leader of my primate group, Scott Muller. And what we did was we took the preformed fibrils and we put them in a striatum of monkeys. And you can see here, a year later, we still have expression of alpha-synuclein within the striatum. But what this also does, not quite as comprehensively as MPTP, but what you can see here, a monkey that received alpha-synuclein preformed fibrils into the nigra, you can see a decrease in both the intensity of tyrosine hydroxylase staining here in panel D compared to controls, or quantitatively the neuronal number of Th positive cells decreases, although not to a level that we really get a tremendous phenotype, but behavioral phenotype. But still, from a pathological point of view, these animals look similar to what you see in Parkinson's disease. And when you look in the striatum, in the substantia nigra of animals that received preformed fibrils, what you can see here are these abnormal cells within the substantia nigra, the spreading of the pathology going from the striatum to the nigra. And also you can see here that these cells are thioflavin positive, and this is actually the first demonstration ever that in a non-human you can have authentic Lewy bodies. And so that's pretty exciting here what this model can give you. So in terms of non-human primate modeling of Parkinson's disease, we have MPTP, we have preformed fibrils. Also, there's wonderful work by Oren Bazard and Jose Obeso using Lewy body extracts. And so these are wonderful models to be using for the study of neurodegenerative diseases. So now I want to switch to a separate disease, and that is multiple system atrophy. I'm just going to briefly.
The chair is falling over. The girl is on the floor, assisting him. Just looking. One is washing the dishes.
Oops. Well, that previous slide didn't play, but what I wanted to show you there was a different disease called multiple system atrophy, which is an atypical Parkinsonian disorder. Here, the synucleinopathy occurs in oligodendroglial cells. What I tried to show you on the video that didn't play is that MSA is like Parkinson's disease on steroids. Patients only live between 7 and 9 years post-diagnosis, and it is just an absolutely horrible disease to have where they have no treatments whatsoever. Maybe a little effects on levodopa early on, but really not much thereafter. These are my students, David Marmion and Diptaman Chatterjee, who performed these studies. And what we found was we had a viral vector that we injected into the striatum of monkeys here, and you can see that there's this OLIG2 marker for oligodendroglial cells, and you can see all the cells here that have GFP also have OLIG2 here in the merged image. Very few cells are neuronal, very few cells are astrocytic. And then when you put alpha-synuclein on this OLIG1 vector, you can express alpha-synuclein into the striatum here in the putamen in going rostrally to caudally. Here standing for LB509, which is a pan-synuclein marker, but it also stands for serine 129. And then what I want to show you, I'm just going to go through the quantification down here, is that you get loss of cells in the striatum, loss of cells in the substantia nigra, increases of inflammation, which HLA-DR, increase in striatal GFAP, and increase in striatal CD3. So all these markers are similar to what you see in multiple system atrophy. And then if you summarize all the things that we looked at, if you look at alpha-synuclein in non-human primate at 3 months, at 6 months, and human MSA, virtually all these markers, PK resistance, I'm not going to go through them for brevity, but all these T cell markers are, similar between the monkey model and the human model. So this is really important in terms of establishing new therapies using this model for patients that have an incredibly rare but serious disease, fatal disease, and require therapeutic interventions. Now we have a model we can really test. There's also large animal models of Huntington's disease. This is work very often done by my former student, actually my always student, Jordan McBride. And I don't have time to go into all this, but there's lots of advantages to studying non-human primates for Huntington's disease. And based upon these studies, there's actually clinical trials going down trying to lower mutant Huntington in patients with Huntington's disease. So I'm going to skip this. So finally, I just want to talk in the last few minutes about this tau monkey model that we're working on. This is work with John Morrison. We're injecting P320F and P301L mutant tau injected to the monkey entorhinal cortex, sacrificed 3 and 6 months later. You can see the imaging here, how we did our surgeries. In terms of biomarkers, following these surgeries, neurofilament light chain, total tau is increased, BDNF is decreased, just like what is seen in Alzheimer's disease. And then there are all these other isoforms of tau that are found in the CSF and plasma of these animals. But fascinatingly, when we inject into the entorhinal cortex, you can see red here, which is AT8 staining, which is a pretangle. At 3 months, you see mostly red. At 6 months, you see mostly green, which is thioflavin S. So you have an evolution between at 3 months and 6 months, which is a time window for which you can use for therapeutic intervention. If you look at the hippocampus of these animals, you can see at 3 months, you see almost all the cells in the hippocampus are AT8 positive, so they're pretangles. By 6 months, you can see the emergence of green, which are full neurofibrillary tangles. We can also see this on with tau staining, tau imaging. So this is a very robust effect. We also, at 6 months, but not 3 months, we have a loss of cells in the CA3 hilus region, loss of cells in CA1, and a trend, but because we have small numbers, about 4 per animal, in the subiculum and entorhinal cortex. And we also have shrinkage of the hippocampus on the injected side, very much what you see in the Alzheimer's disease. And if you look at all these regions between 3 and 6 months, looking at pretangle, mature tangles, and ghost tangles, you can see the evolution of the pathology that's seen here. And then finally, we know, and I'm not going to go into detail about this, but there is this is all permissive templating because the vector itself only encodes for 4R tau, but when you get away from the entorhinal cortex, such as the hippocampus, you can see the AT8 co-ops with 3R tau. So this is permissive templating, the spreading that we're seeing around the brain. And so we now have demonstrated, following injections into the entorhinal cortex, we get spreading to the hippocampus, retrosplenial cortex, visual cortex, and also not shown here, we have it in temporal neocortex. So final thoughts. It's important to employ non-human primates to study human diseases, although not every question needs to be tested in monkeys. I should say monkeys are not humans. It's important to employ the 3 R's, replacement, reduction, and refinement, how these concepts are only applied when you still can adequately test the hypothesis at hand. The development of new primate models are emerging in multiple domains, and since ADPD and other degenerative diseases contain multiple pathologies, such as misfolded tau, amyloid, alpha-synuclein, TDP-43, non-human primate models of co-pathologies should be included when you're thinking about advancing modeling. And this is the group that I work with, and I'm very proud to have them in my lab. And with that, thank you.
Okay, thank you very much, Dr. Cordova. I'd like to remind everyone to please put your questions into the chat so that at the end of the session we can have a nice discussion. Our second presentation is from Dr. Azadeh Yazdan Shamarad, Assistant Professor at the Washington National Primate Research Center, and the title of her presentation is Engineering Cortical Plasticity in Non-Human Primates Using Optogenetics.
Hello, everyone. Thanks for having me here and for the opportunity to talk about my research. I started my lab in 2017 at the University of Washington. And in my lab, we build novel neural technologies and hope to use them to develop therapies for neurological disorders. In this talk, I'm going to talk about some of those technologies and how we use them towards targeted reorganization of the brain. And the goal is to use these technologies to develop therapies for neurological disorders such as stroke. So the reason we're focusing on stroke is because, as you might know, stroke is one of the leading causes of long-term disability in the United States, and current treatments provide only modest benefits. Over the past couple of decades, people have been using neuromodulation as a therapy for stroke, techniques such as magnetic stimulation and electrical stimulation. However, the effectiveness of these techniques has been a matter of debate. You might be familiar with the failed Everest clinical trial, which I'm showing some figures here. And the goal was to combine epidural cortical stimulation with rehabilitation to accelerate recovery from stroke. However, the results failed to show efficacy for the patients who received stimulation in comparison to the patients who did not receive stimulation. And I actually was involved in this clinical trial as a part of my PhD. And from my perspective, some of the reasons for the failed clinical trial, as well as some concerns that exist regarding using neuromodulation for stroke therapy, is due to the fact that for the majority of these cases, stimulation is not specific and affects large regions of the brain. Also, again, the majority of them, the stimulation has been open loop, meaning that the dynamics of neural activity and behavior were not incorporated in generating stimulation protocols. And lastly, we don't have a good understanding of the underlying mechanisms of stimulation-induced plasticity. So as you might know, optogenetics is a new neuromodulation technology that has several advantages over the previous neuromodulation techniques and can address some of these challenges and provide insight about these mechanisms of plasticity and stimulation on the brain. So for those of you who might not be familiar with optogenetics, briefly, there are these light-sensitive ion channels that come from certain type of algae. We can genetically modify neurons and place these channels on the surface of the neuron. So by shining light, we can control the activity of the neuron, activate them or deactivate them. So some of the advantages of optogenetics are cell type specificity. We can do this cell type specificity control with high spatial and temporal resolution. We can do inhibition as well as excitation. So we could do multicolor optogenetics. And also we can have artifact-free recording, which is important because then we can investigate the direct effects of stimulation. Also, it opens up more opportunities for closed loop or activity-dependent stimulation. All of these makes optogenetics a powerful tool to study the brain and its networks. And if we combine that with behavioral capabilities of non-human primates, we might be able to answer fundamental questions about the brain function or to develop novel therapies for neurological disorders. During my postdoc, I developed a large-scale optogenetic interface for non-human primates by combining 3 technologies. So I'm going to go through each of the technologies. The first one was a novel viral delivery technique that was called convection enhanced delivery and was based on pressure gradient. This technique enabled us to achieve high levels of expression across large brain areas. We co-infused the virus with an MR contrast agent, and we were able to monitor the infusion during infusion. And then we also, here you see histology that confirmed large expression. this technique provided orders of magnitude improvement over other viral delivery techniques in the area of coverage and the time that it takes to deliver the virus. So the second technology we used was artificial dura. This is a clear silicone-based material that provides optical access to the brain. That optical access can be used for light stimulation, but also for imaging. Here I'm showing in vivo validation of our large-scale expression. Here, as you can see, we imaged the fluorescent reporter of our virus. This is a virus that we use that was targeting excitatory cells, and it was expressing an excitatory channel, meaning that by shining light, we were exciting the underlying network. Lastly, we used microelectrocorticography or micro-ECOG arrays. These arrays were developed to be semi-transparent, so they're sitting on the surface of the brain, providing recording from the underlying expressing network, and then we could place our fiber optics laser light sources anywhere in this area and control the activity of the brain. This interface provided a very nice platform for us to study, can we use optogenetics to change connectivity in primate cortex? We designed our experiments based on the Hebbian learning model. We had a very simple stimulation paradigm. We either had our laser or fiber optic in primary motor cortex or M1, or in primary somatosensory cortex or S1. We had 10-minute blocks of conditioning that were interleaved with baseline recordings that enabled us to evaluate the effects of stimulation. So let's see what happened. Here I'm showing the initial functional connectivity maps. So the blue location shows the location of stimulation, the blue circle, and the diameter of these other red circles show the strength of the connectivity between that location with the site of stimulation, which is shown in blue. So these are two examples of initial functional connectivity maps. And then after 15 minutes of stimulation, the strength of functional connectivity are shown with these white circles. And as you can see in these two examples and in our summary plot, which is across experiments and animals, we were able to significantly strengthen functional connectivity between these two brain areas. So this result was very exciting because for the first time, we showed that using optogenetics, we can change functional connectivity, we can strengthen connectivity between two brain areas. However, as you can see here, this is at a gross level, right? I'm showing that between motor and sensory cortex, we are strengthening the connections. But I have about 100 electrodes that I'm recording from these areas. And if I want to understand what is happening between each pairs of electrode, then that was a more complicated question. However, if you remember, initially I talked one of the advantages of optogenetics is that we can record during stimulation. So we tried to understand what is happening when we're stimulating the brain. And we generated these maps where each of these maps shows the coherence of that electrode with all of the other electrodes during stimulation. So this map is showing us this is what's happening between all the electrodes during stimulation. And then we generated a similar map on how the network changed following stimulation. And the remarkable result we found is that if you compare these two maps, or if you compare the color patterns of these larger maps, they look very similar. And indeed, we found a linear relationship between them that was consistent across animals and experiments. So this is showing that during stimulation, we're forcing the network to fire in this particular way, and it ends up being wired in that way, which is consistent with the Hebbian learning model. Now, this is, as I said, was a very simple stimulation paradigm. We had one laser either in motor or in sensory cortex, but we tried to use more complicated patterns of stimulation. So we added more lasers and we changed the delay between the lasers. And we saw that no matter how complicated our stimulation paradigm, as long as we knew what was going on during stimulation, we could predict how the network was changing at a fine scale. So this was also encouraging, but you could tell me that, okay, if I want to change the network in this particular way, what should be my stimulation paradigm? And this is a question that Julian Block, who's a PhD student in my lab, has been trying to answer in the past couple of years. And he's been working with mathematicians and statisticians at UW to build hierarchically penalized additive models to understand this. And so his model is taking in information about both stimulation protocol information as well as information from the underlying network, in particular the functional connectivity of the underlying network. And as I said, this is a model that enables nonlinearity between these features and the functional connectivity. And so so I'm showing some examples of these functions that were extracted from this model. And I'm not going to go through those, but what I want to highlight here is that one, his model is performing very well. It has R squares of 0.4, but also what I want to also emphasize here is that we see that underlying network features, functional connectivity of the network has a much bigger contribution to the connectivity changes in comparison to the stimulation protocol features. And to give you an example, here is an example of the actual connectivity change that we're seeing. And if we are using his model, if you're inputting his model only protocol features, the model is doing a poor job in predicting how the network is changing. However, if we are combining the stimulation protocol with network features, then we get a much closer prediction and a much better prediction of the changes. And so this result is showing that the underlying network, the functional connectivity of the underlying network plays a big role on how the network responds to stimulation and how we are changing the network. And so it shows that, for example, if we have a stroke patient coming to the clinic, we have to tailor our stimulation to the underlying functional connectivity of the network. That could be different depending on the size and location of the stroke, for example. And so we have to incorporate all of that information into our stimulation protocol and, in a way, personalize our stimulation protocol. So what I've shown you so far has been on excitatory optogenetics, right? But then initially I talked about inhibition as well. And one question would be, can we change the network with inhibition? Because when we combine them, then we can potentially create like more complicated patterns. Okay, so to do this, we have been trying to implement that over the past couple of years, and as a part of that, we have been optimizing our large-scale interface. So I'm going to take a few minutes and talk about how we have been optimizing our interface. We worked with Ripple Neuro to print platinum particles into our artificial dura, and now we're calling it multimodal artificial dura. Another cool thing about this multimodal artificial dura is that, as you see here, there are no PCBs here. And so that enables us to store these arms in our chamber when we are not recording. And then when we want to record, we can clamp our PCBs onto these connectors. And this way we can protect our PCBs from corrosion. And another advancement that we have done in comparison to the previous version is that we have built LED arrays that now enable 16 independent locations of stimulation simultaneously. We have built them in different colors, and then they are compatible with our interface and our multimodal artificial dura, so they can sit on top and illuminate with different patterns. So we have implemented this interface into monkeys. I'm going to show data from one animal. As I said, this is an inhibitory opsin. We use JAWS, and here you can see large expression from our epifluorescent imaging. We see deactivation of the network from neurophysiology. This is implanted in posterior parietal cortex, which is involved in multisensory integration, and lesions in PPC have shown to impact reach and grasp. So in a center outreach task, we are showing that whenever we are deactivating PPC using inhibitory optogenetics, we are increasing the time of the reach. And so this is also a very exciting preliminary behavioral results that we have, that to my knowledge, this is the very first behavioral correlation with optogenetics in the context of a reach task. But more importantly, going back to our question of can we use optogenetic inhibition to change connectivity. And so our initial analysis of our data from one animal says yes. So here I'm showing data during stimulation that shows that during stimulation, we're decoupling the site of stimulation from the rest of the network, but that change persists after we stop the stimulation and we see some plasticity. So basically we're showing that we're weakening the connection between the site of stimulation with the rest of the network. And if you remember, what I showed from excitatory optogenetics was that we were strengthening connection between the site of stimulation and the network with excitatory optogenetics. And so this is showing that we can combine these two modalities of excitation and inhibition and create like more sophisticated patterns of stimulation and have like more detailed reorganization of the brain with multicolor optogenetics. And so the inhibitory optogenetics, excitatory optogenetics, in combination with our model that I showed you before, paved the path for us to reorganize the brain towards targeted functional recovery from neurological disorders such as stroke, right? But I said functional recovery and I said stroke, but everything I've shown you so far has been in a healthy, normal brain. And we have been working on incorporating models of stroke into our interface because ideally you would want to study that in the presence of an injury to be able to monitor functional recovery. So this is a work by Karam Khatib in the lab, which is based on the photothrombotic technique. This is a photochemical lesioning. So you inject a dye IV. This dye is sensitive to light, and it will be everywhere in the bloodstreams. And then anywhere that you shine light, you can trigger the coagulation cascade that forms thrombi and then create an ischemic lesion. So we implemented this in 7 macaques. Here is an example of the surface of the cortex. We collaborated with Ricky Wang at UW to image blood flow. So here's using optical coherence tomography and geography. Then we injected the light-sensitive dye, and then imaged and then illuminated those areas. And then after 4 hours of illumination, we were able to see that the blood flow was stopped to the areas that were illuminated and could create focal lesions. These were validated through histology and showed that we have these focal cell death and neuronal death. We also use our multimodal artificial dura and created these lesions through that our MMAD, enabling us to record neurophysiology, neural activity as the stroke was forming. So here I'm showing an example from a monkey. This is the baseline recording, and this is the change in activity after stroke. So this is the area of illumination. The activity went down. See the change, and if we categorize them, we see areas that show a decrease in activity that's close to our illumination, close to the lesion, and then we have areas that the activity didn't change, and we have these blue areas that the activity increased. And we saw the same pattern for our second animal. And if we use this color coding for the areas that change, we can actually follow and monitor how each of these activities were changing as the stroke was forming. So for example, here you see that everything goes down, but these red electrodes, even after the illumination, stay down because of the cell death, neuronal death we're seeing following stroke. But then the rest of the network goes back to normal, and even maybe we see some compensatory mechanism. But it shows you the capability of our interface. I want to remind you that this can be done in the same paradigm of our optogenetic interface. So basically, we can have inhibitory optogenetics, excitatory optogenetics. We can induce focal lesions with that, and we can combine this with behavioral capabilities of non-human primates and monitor functional recovery. So as you can imagine, the combination of all of these technologies provide a lot of potentials for us to explore targeted reorganization of the brain that can be used as a therapy for stroke and other neurological and psychiatric disorders. With that, I want to thank all of the amazing people in my lab, people who help with animal care, our collaborators, our funding agencies. None of this would have been possible without the help and cooperation of our monkeys. And before I end, I want to say that we in the lab have a postdoctoral position that is supported by a recently funded NIH R01. And so through this position, not only there's a lot of opportunities to work with all these cool technologies and study functional recovery from stroke, but also we have a very fun and welcoming lab culture. We value kindness and growth, and based on those values, we try to create an equitable and inclusive environment for everyone. So if you're interested, please contact me. This is my email. And if you don't mind, please spread the word. Thank you so much.
Thank you very much, Dr. Yazdan Shamaran. Our third and final speaker is Dr. Jody McBride, Associate Professor of Neuroscience at the Oregon National Primate Research Center. And the title of her talk is Developing a Rhesus Macaque Model of the Degenerative Brain Disorder, Huntington's Disease.
Good afternoon, everyone. I'm Jody McBride. I am an Associate Professor of Neuroscience at the Oregon National Primate Research Center. I'd like to thank the organizers for the invitation to speak today and to share data on behalf of my laboratory, as well as all of our collaborative partners at the ONPRC. So the focus of my lab over the past 10 years or so has been evaluating gene therapy strategies for different neurodegenerative brain disorders. And we primarily focus on Huntington's disease and Batten disease. In part of doing that work, through developing therapeutic strategies, we realized the critical importance of having appropriate large animal models to work in, more specifically, appropriate non-human primate models that will better model the neuropathology and behavioral outcomes that we see in patients with these disorders. Now, a part of that work, we've used novel approaches to try to get genes delivered into the central nervous system, by example, using novel viral vectors, different capsid variants, different delivery strategies, and I'm going to focus on some of those today. So for those who are not familiar with Huntington's disease, or HD, this is a fatal neurodegenerative brain disease, and it's caused by a mutation in exon 1 of the Huntington gene. The mutation was described in 1993 by the HD Collaborative Research Group, and the mutation itself is a CAG expansion. So in the DNA bases C, A, and G that encode for the amino acid glutamine. Normally, Huntington has fewer than 36 of these CAG repeats, and in Huntington's disease, the gene has more than 36 to 40 repeats, giving rise to a Huntington protein that has an abnormally long stretch of glutamine residues at the N terminus. Now, because of this, the protein misfolds, and due to this altered conformation, it's not able to be processed appropriately by the ubiquitin proteasome pathway. And like we heard in an earlier talk in this session, those misfolded proteins then tend to clump together and form inclusions or aggregates that can be stained, as I'm showing here. So historically, the pathology of Huntington's disease was described in postmortem brain tissue from affected patients. And the primary brain regions affected in Huntington's disease are the caudate and the putamen, collectively making up the striatum, as well as the overlaying cortex, but also the white matter tracts that connect the cortex down into the caudate and putamen are also affected in this disease. And then, of course, you can see in postmortem samples also examples of the mutant Huntington aggregates. So currently, HD pathology is typically tracked in the clinic using MR imaging. So you can see here in a scan from an HD patient on the left, that really profound atrophy, especially of the caudate and putamen. as well as the overlaying cortex. This is used throughout the clinic as well as in clinical trials as primary outcome measures. But more recently, there's been a big push in the field to develop biofluid biomarkers of disease progression. And so there's been a handful of studies published over the last several years showing that there are disease-related proteins that we can detect in the CSF, including mutant huntingtin itself, as well as the protein neurofilament, among several others. So this is a little bit of a complicated slide, but I want the take-home message to be that Huntington's disease is not a disorder of just one or two brain regions. It really is a disease of circuit disruption that involves lots of different cortical areas and lots of different subcortical areas that give rise to this complex constellation of symptoms that patients experience. And so shown here in left, in red, patients show cell loss up in motor cortical areas that drive down into and throughout the basal ganglia. And because of cell loss in this circuit, patients experience motor symptoms early on as the loss of fine motor skills. This progresses into uncoordinated movements and hyperkinetic involuntary movements that are known as chorea. And here I'm showing you a patient that is seen in the OHSU Movement Disorders Clinic here. And I hope that you can appreciate, I'll play this again, those uncoordinated movements and the hyperkinetic movements that you can see in the upper extremities. In addition to motor symptoms, patients also show cognitive symptoms. That's due to loss of cells up in these prefrontal areas shown in green, down through the basal ganglia, working memory decline, attentional deficits, and impulsivity can be common in Huntington's disease. And then in the limbic loop, because of cell loss in these limbic areas that project down into limbic areas of the basal ganglia, cell loss in these areas is thought to lead to some of the psychiatric symptoms in HD patients like anxiety and depression, aggression, and also often perseverative behaviors. And so again, the take-home message is that this is a complex disease that involves many circuits. It makes it complex to treat, and it also makes it complex to model. And unfortunately, there are no therapies that prevent Huntington's disease. There are a few treatments available aimed at targeting specific symptoms. And so, for example, anti-chorea medications are aimed at decreasing the neurotransmitter dopamine. And in fact, tetrabenazine is the only FDA-approved drug to treat Huntington's. Well, now deutetrabenazine has been a second modification of tetrabenazine that was more recently approved. And these aim at reducing the amount of dopamine that is available in the synaptic cleft between these two neurons. Unfortunately, for mood and cognitive symptoms, there are no FDA-approved drugs specific for Huntington's, but patients are often prescribed antidepressants and anxiolytics for the mood symptoms. Unfortunately, for cognitive symptoms, there really is no available treatment, and the focus is on modifying the environment of the patient. So over the past several years, there have been many animal models of HD under development, ranging from the worm and the fruit fly all the way up to the larger animal models. As you might imagine, the majority of research has been in transgenic and knock-in mouse models. That's really been the mainstay in the field. However, the last decade has really seen the push for these larger animal models in the sheep, pigs, and non-human primates really mandated by the FDA, because we've had a lot of examples similar to other fields as well, where therapies were screened in rodent models, moved directly into clinical trials, where unfortunately they didn't succeed. And so now there's a big push to be evaluating these therapeutics in rodent models first, but then very quickly partnering with those that have the larger animal models to test these therapies before going into clinical trials. There are large animal models in development all over the world, I'm happy to report. I won't have time to go into each one of them, but to show the progress that's being made over here on the left, and specific to the non-human primate, there's great progress being made in several different institutions. For example, Guoping Feng's group at MIT is working on a CRISPR-Cas9 based knock-in marmoset model that's still at the cellular stage. Here at the OMPRC, John Hennebold also working on the same strategy in the rhesus macaque. And then what I'm going to talk to you about today is work that my lab is doing using adeno-associated virus to deliver mutant huntingtin to make an AAV-based model in adult macaques. So when thinking about creating a model of Huntington's disease in the macaque, we really wanted to develop a model of early stage HD. These are the patients that are going into these phase 1 and 2 clinical trials. And so we wanted to create an appropriate animal model that would allow us to partner with folks to screen these therapies in the same stage that will be used in the clinic. So we wanted to induce a progressive motor and cognitive decline, more on the mild, and also progressive atrophy of white matter tracts and gray matter regions throughout those cortical basal ganglia circuits that I just showed. Of course, inducing hallmark neuropathology of mutant huntingtin aggregates. Also signatures of altered neurotransmission. We were specifically interested in dopamine, which I'll talk about further, and mitochondrial dysfunction. And it was really important for us to create a model that's accessible to the HD research community. And what we meant by that was to think about a model that we could create in sufficient numbers that we could power future biomarker discovery studies and future therapeutic studies, and be able to create animals sort of quickly and in large enough numbers to make those studies feasible. And so for that, we decided that AAV-based modeling was the way to go. At the OMPRC, the way that we deliver genes into the non-human primate CNS is typically with AAV vectors. Historically, we've worked with different types of AAVs, including 1, 9, and 2, also some capsid variants, namely PHPB and 2 retro. And today I'm going to talk about work that we've done with AAV2 and AAV2 retro. There's different ways that we can deliver genes into the CNS. We primarily use intravascular infusions into the cerebral spinal fluid. But today I'm going to be talking about direct brain injection, so lowering a cannula deep into the brain, directly into brain parenchyma. So a little bit about 2 retro. So we became really excited about AAV2 retro when this paper was published in 2016. And this is a joint effort by Alla Karpova's group at HHMI and David Schaefer's group at Berkeley. And through a directed evolution approach, they identified AAV2 retro that has really potent retrograde capabilities when injected into the central nervous system. And what that means is that when you lower a needle into a region of brain and deliver these particles, they get taken up at the axon terminal, brought back down through long axons, sometimes several centimeters long, back to the cell body where they express a particular transgene of interest. All of their work was initially done in mouse models. And so here I'm just showing an example where they used AAV2 retro to express 3 different transgenes of different colors that can be stained in the brain. And I hope you can appreciate the really beautiful expression pattern given by this AAV2 retro. So not only do we see cell bodies throughout the mouse striatum, but we also see cell bodies all throughout the mouse cortex, particularly in those regions that are of interest to us for Huntington's, up here in those frontal cortical areas, back in the motor cortex, as well as some subcortical areas, of course, the striatum itself, but also areas like the amygdala and the thalamus. So we were really excited to try this vector. We got the plasmid sent to us from their group. They also have this on Adgene for those that are interested, and conducted a short pilot study to try to replicate their data in mice. So we delivered AAV2 or 2 retro, expressing eGFP bilaterally into the caudate and the putamen. And then 4 weeks post-surgery, took a look at vector expression. So here we're looking at GFP staining. And what we found was that with the parent serotype AAV2, we got really beautiful transduction where we injected the vector in the caudate and putamen all throughout, but not really much appreciable transport to any cortical areas. And in contrast to retro, showed expression in the areas of injection, but also just beautiful transport back to all of these cortical areas. Just really nice. Anywhere you see this purple ribbon is cells expressing eGFP. And so armed with this knowledge, we wanted to move forward then expressing Huntingtin, our gene of interest in modeling Huntington's disease. So for this next study, we delivered a fragment of human Huntingtin expressing an expanded CAG tract, so 85 CAGs, or for the control 10Q. And what we found is now you're looking at staining 10 weeks post-surgery, and we found that we saw mutant huntingtin inclusions in all of the same brain regions that we had seen in the EGFP pilot study. So with this confirmation that we could deliver mutant huntingtin and induce those hallmark aggregates, we set out to do a much longer study. So in the long-term study, we had 3 groups of animals that received either just a phosphate buffered saline, animals that received the control fragment, or the pathogenic fragment. These animals were trained at baseline on a variety of behavioral tasks. And then we also collected multimodal imaging and biofluid. And then post-surgery, we collected those same assays at either 3, 6, 9, or 14 months. And then we were out of lab a little bit for COVID, but then came back strong. And I'm going to show data now out to 20 months. And around this time, we also had the Primate Multimodal Imaging Center come online here at the West Campus. And so I'll show data from some new PET scans here out at 20 months post-surgery. And so just one note about the difference of surgery here. We used a little bit of a Goldilocks approach where we decided to mix AAV2 and AAV2 retro expressing mutant huntingtin or control huntingtin, because we felt that 2 retro was so efficient at transporting that not enough was staying in the caudate and putamen. And so by mixing these 2 together, we were able to fill the caudate and putamen, but then also get really nice transport throughout all these other regions of interest. Again, these were MRI-guided surgeries, bilateral injections. So the first thing that I'll show you is some motor behavior. So to query the function of motor circuitry, we used an NHP neurological rating scale. We modeled this after the Unified Huntington's Disease Rating Scale that is used in the clinic to rate human HD patients. This is a score that ranges from 0 to 3. Animals are given a score of 0 for normal behavior, up to 3 for completely abnormal behavior on a variety of different measures. And so these are summed together, and higher scores equal aberrant motor behavior. The 85Q animals shown in orange, you could see this progressive development of aberrant motor behaviors over time that began around 3 months of age. And I've highlighted with a little yellow flag on the left those behaviors that we noticed in these animals. We then did an apomorphine challenge. Apomorphine is a dopamine receptor agonist. And so we rated these animals before and after apomorphine and found that this exacerbated some of these behaviors, suggesting to us that some of the dysfunction that we're seeing is due in part, at least, to dopamine dysregulation. Okay, we also looked at working memory decline in these animals, and we used a 3-choice delayed response task to do so. And the details of the testing are shown under the photomicrograph on the left. So again, with 85Q treated animals in orange compared to our 2 control groups in blue and gray, we see that animals are making more errors. So a well is baited with a food treat or a candy treat, and then the animals are asked to recall which well was baited after variable delay, and animals are showing more errors treated with 85Q. And this is just that data collapsed over time. Okay, so now I'm going to switch to some of our imaging findings. So we're really lucky here at the OMPRC to have wonderful imaging facilities. The MRI core is directed by Dr. Chris Kroenke, and the new PIMIC is directed by Jonathan Lindner. And so I'm going to be showing both MRI and PET data to you all. Okay, so to first look at whether or not we had any regions of gray matter atrophy, we use something called tensor-based morphometry. And what I'm showing here are T2 weighted MR image templates with the buffer group on top, our 10Q control here in the center, and the pathogenic 85Q animal shown here on the bottom row. And so what you're seeing is anywhere that I'm showing a blue voxel is an area of significant tissue contraction compared to baseline. So this is showing degenerative gray matter changes. And here I'm just plotting this as the p-value of that change. And to show which brain regions showed the most dramatic changes, the bar graph here is showing us that the putamen, the caudate, as well as the globus pallidus, and a variety of frontal, a whole bunch of prefrontal and motor areas are showing the primary areas of tissue contraction or atrophy. And then similarly, to look at white matter, those white matter regions that connect the cortex down to specific other cortical areas or down to the basal ganglia is shown here. So compared to buffer and 10Q controlled animals, here I'm showing you the fractional anisotropy templates, and the blue colored voxels here in the 85Q treated animals are showing anywhere where we had reduced fractional anisotropy compared to baseline rather. And during degenerative disease processes, as those white matter fibers start to show microstructural changes, we get lower levels of FA indicating fiber damage. And the white matter tracts that showed the most profound changes were ventral prefrontal white matter tracts and the corona radiata, which connect those prefrontal and motor areas down into the caudate and putamen. Okay, so for the final minutes, I'm just going to switch over to our PET imaging studies. So this is a cartoon showing all of the ways that mutant huntingtin wreaks havoc inside the cell, and it does so in many different ways. The ones I'm going to focus on that we were interested in looking at with PET scanning are looking at altered protein folding, trying to look at those mutant huntingtin aggregates, looking at disrupted circuitry. So we used F18 phallipride to look at dopamine receptor binding. We also did FDG PET scanning that I'm not going to have time to show today. Okay, so the first that I'm going to show you is phallipride imaging at 20 weeks post-surgery. So you're looking at group averaged phallipride PET images showing D2, D3 receptor binding potential throughout the brain. And binding potential was calculated using the Logan reference tissue model and using the cerebellum as the reference region. I hope you can appreciate that the 85Q treated animals are showing lower binding potential or less red compared to 10Q and buffer controls. And we can see that data down here quantified in the bar graph. We see reduced dopamine binding potential in the caudate and putamen, as well as the external globus pallidus. Okay, so the last thing I'll switch to is newer work that we've done in the lab, looking at the novel CHDI radio tracer CHDI 180R. This is a new tracer that was developed by the CHDI Foundation. And first I'm going to show you beautiful work that was done in an HD mouse model. This work was done by Danielle Bertoglio from the University of Antwerp, who is a colleague of ours. And what you can see here is compared to wild type mice, now we're looking at the age of mice. And compared to wild type mice, heterozygous HD mice show the development of increased VT over time. So all of these red voxels indicate the presence of mutant huntingtin aggregates. You can see this especially in the cortex and down here in the dorsolateral striatum. And so this beautiful work was recently published. They also corroborated the PET data with immunohistochemistry after those mice were taken to necropsy. So here you can see an increase in the number and density of these inclusions over the lifespan of the animal. So we then worked with CHDI and Daniela to bring this work into our animal model. And so what I'm showing here with the MR template that we used on the left, compared to buffer and 10Q controls, we see the presence of huntingtin aggregates in the caudate and putamen, as well as certain cortical areas. We also noted some staining or some PET signal rather in also white matter tracts connecting these brain areas, which is a novel, interesting finding. And so we were really happy to be able to do this work in the non-human primate to help bring this novel radio tracer into clinical studies. And so I'll very quickly just say that in addition to behavior and PET imaging, we also collected CSF from these animals every 3 months. And these samples have been sent to Evotec for immunoassays to look at mutant huntingtin and neurofilament, as well as plasma levels of neurofilament. We also have an ongoing collaboration with UBC and the NRC in Canada, who's using a targeted mass spec approach to look at about 25 different disease-related proteins in this model. So data to be forthcoming. So I'll just summarize by saying that at the OMPRC, we have developed a novel macaque model of early stage HD that develops several of the cardinal features that we see in human patients. We have a small, short, pre-symptomatic time period, and then followed by several months of what we're characterizing as early symptomatic changes in these animals. These animals went out to 36 months and have gone to necropsy, and we're in the process of looking at these tissues using a variety of histological and molecular techniques to corroborate the imaging findings. So I'd like to acknowledge the large number of people that worked on these studies, with a particular acknowledgement to Allison Weiss and Will Agor in my laboratory, Chris Cronke, who worked on the MRI studies with us, Jeannie Link, who collaborated with us on the PET studies, and of course, everybody else that was so instrumental to the success of this work. These studies were funded by the NINDS, the Bev Hardig Huntington's Disease Foundation, as well as the CHDI Foundation. And I'll just end with this slide. This is sort of a summary of manuscripts that have come out of all this work. And if anyone is interested in learning more about any of these, I've created some QR codes. So please, if you're interested in learning more about large animal models, the pig and the sheep compared to the monkeys, please check out our review on the large animal models of HD. We created a novel macaque brain atlas to complete this work. So if you're in the neuroscience space and you're interested in that atlas, please see this QR code here that links you to the new atlas. If you're interested in learning about AAV2 retro-transport throughout the brain, either for modeling or for therapeutic development, please visit this manuscript. And if you're interested in the new model that I talked about today, we've just uploaded a brand new paper to bioRxiv. So thank you so much for your attention, and I'll stop there.
Thank you very much, Dr. McBride. We can now start the question and answer session, which is live. For the first question is from Dr. Matt Arnegard, which is for Dr. Cordova. Can you do lateralized control technical approaches in the same animals with AAV, AD monkey model with lateralized behavioral readouts?
We cannot do that. I mean, we can do that, but we cannot do it rigorously because the spreading occurs bilaterally. So when I didn't really mention it, but when you put the AV double mutant SAL into the left entorhinal cortex, it will transport or permissively template across to the contralateral entorhinal cortex and other regions in the contralateral hemisphere. So that would be a very difficult task to do and be able to interpret.
Thank you. And our next question is from Allison Weiss, also for Dr. Cordova. If the AAV2 tau NHPs have any cognitive or behavioral deficits, if unknown, are there plans to assess cognition in these monkeys?
Super excited about this model, and we applied to NIH for an R24 application, which we got. And one of the aims in that application, being led by Mark Baxter from Mount Sinai, is to do cognitive testing in bilaterally treated animals and controls. And we're going to do delayed non-match to sample, and we're going to do scene memory testing because both of them are responsive to perturbations in the entorhinal cortex hippocampal axis.
Thank you. And again, Dr. Cordova from Dr. Ron Veazey, how does the rhesus and marmoset model compare for neuroscience studies?
Well, marmosets are better than rats, but they're small non-human primates. They do have a bundled internal capsule, but still their cortical physiology and cortical anatomy is not the same as a rhesus monkey. Also, if you're doing studies for therapeutics, it's more difficult to estimate dose in a marmoset for human use than it is for rhesus monkeys. For those reasons, I don't use marmosets at all, and I will use cynomolgus or rhesus monkeys exclusively.
Great, thank you. The next question is from Dr. Michelle Basso for Dr. McBride. Was the substantia nigra labeled?
We get this question a lot. Thank you for asking. In the short-term studies that we did where we delivered AAV2 retro with either GFP or the follow-up study where we delivered mutant Huntington and took the animals to necropsy after either 4 or 10 weeks, we saw transport back to substantia nigra. I will say this was confirmed anatomically, right? So we were basing this off of either GFP staining or mutant huntingtin inclusion staining in the substantia nigra itself. We didn't do follow-up studies, for example, double label studies with mutant huntingtin and tyrosine hydroxylase to confirm that they were in fact dopaminergic neurons. But given the reception that we've got in shopping this data around, there's a lot of people trying to get too retro back to the substantia nigra. So in this larger cohort of animals that have gone out 36 months, we're definitely going to do some more diligent double labeling of the nigra so that we can help answer those questions.
Great, thank you. Another question for Dr. McBride from Thomas Wickman. Regarding HD models, what translational experiments would you plan next now that the HD model has been characterized?
Yeah, so lots to think of there. In the HD space, a big push now is Huntingtin lowering therapeutics. In fact, the small handful of studies that have reached the clinic currently are all Huntingtin lowering, or the majority of them, I should say. So now that we have this model, there's some considerations there. Because this model is vector-based, I would say that testing therapeutics that are not vector-based would be the first priority. And one of the biggest pushes now is antisense oligonucleotides that can be delivered either intravenously into the CSF, and that can target mutant huntingtin. So we're excited we've created a partnership with some colleagues up at UBC that have huntingtin lowering antisense oligonucleotides. So that would be the first place to start. But we have interest in trying to do those assessments where we use AAV-based therapeutics in this model, and look at how successful a second AAV administration would be after the first to create the model.
Since the CHDI 180R ligand binds to the HD, can you use that therapeutically?
I can't use it therapeutically. It doesn't do anything to lower huntingtin expression, but we're super excited that it's just entered the clinical space to be evaluated in a small handful of Huntington's patients. So we recently were at a conference where they showed data from the first 3 Huntington's patients that received PET scanning, and preliminary data looks like it's efficacious in humans. So it will be a great imaging biomarker for these Huntingtin lowering trials that are in the pipeline.
Yeah, that all sounds good, but since it binds the aggregates, maybe there's a way to link it to something else that could cause it to opsonize. I'm just thinking potential. Okay, the next question is for Dr. Cordova. Can you comment on the occurrence of dystonia in your models of Parkinson's disease? And this is also from Thomas Wackman. And are there models or conditions that would show more dystonia than others?
So when you have your monkey models that are synuclein based, you see very little dystonia. You do see dystonia with MPTP, especially if you give it up the carotid artery. You get this crooked arm dystonia and sometimes a leg dystonia as well. If you want to study dystonia, one of the most robust models that Dr. McBride and I published many years ago was with 3-nitropropionic acid. We were actually trying to create a model of Huntington's disease, and we created a model of dystonia that only we were interested in going forward. So if you want to study the phenomenon of dystonia, I would use 3-nitropropionic acid or MPTP, but I'm not sure either one of those is what causes dystonia in humans, those mechanisms.
And I can piggyback onto that if anyone's interested in the Huntington's model, that we don't see very robust dystonia. We see more incidences of chorea and postural changes in distal limbs and a little bit of evidence of bradykinesia, but we didn't observe a lot of dystonia in our model either.
Great, thank you. I'd like to ask a question to Dr. Yazdan Shamarad. You applied the optogenetic approach in the setting of stroke. And I wonder if you might comment to what the potential might be for other disorders such as PTSD, addiction, drug addictions, some things like that. Could you comment on that?
Yes, thank you for the question. So what we have shown, so the initial part of my talk, we were showing that in the healthy brain, we're showing that there's a feasibility for targeted reorganization of the brain using optogenetics. And then later on, we incorporated the model of stroke to show that in the presence of an injury, can we do this reorganization? But a good example of what you mentioned for other, neurological or neuropsychiatric disorders, we recently heard that we're going to get funding for an R01 from NIMH to basically implement all these technologies in prefrontal cortex to study or impact rule-based behavior. And so basically the technology is transferable to different areas of cortex.
Great, thank you very much. I have another question for Dr. Cordova. It seems like alpha-synuclein is pretty bad in its expression, and it expresses a lot in brain's Parkinson's disease. How is alpha-synuclein expressed in the context of aging and inflammation within the aging brain?
So first of all, synuclein is not bad. It's in every cell because it's a synaptic protein. We need synuclein. And in fact, if you were to lower synuclein by over 95%, it would cause a lesion. So you need to be in a sweet spot. Too much synuclein is bad. Too little synuclein is bad. So you need to be in the middle of a sweet spot. So the other part, in terms of normal aging, what happens is, so normally you can't even see synuclein in the pericardia of a adult monkey or human, because as soon as it's made, it shuttled down the axon. And then what we think happens is that synuclein starts to rise in the cell, and that slows axonal transport down to the terminal, and that causes lysosomal dysfunction, the buildup in the cell body. And in normal aging, you get increase in the monomeric alpha-synuclein that's associated with decrease in phenotype, dopaminergic phenotype, not cell loss, but phenotype downregulation. And then, as I show in some slides, sometimes something happens, and you flip over from monomeric synuclein to aggregated synuclein, and that's the pathway towards Parkinson's disease. So in aging, there's an increase in the monomer. In Parkinson's disease, there's an increase in the monomer that eventually becomes aggregated, and that causes the synaptic retraction, and that ultimately causes cell death.
Great, thank you very much. I see a question from Dr. Dada, but it's just, I don't see the actual, here it is. Have you looked into the mitochondrial health? Can drugs can maintain mitochondrial health, protect brain cells? And this is to Dr. McBride.
Yeah, so in Huntington's disease, there is definite mitochondrial dysfunction. This has been known for decades, and the data that I didn't get to show in my presentation was that we have verified mitochondrial dysfunction in our model. And so, of course, looking back at the history of therapeutic development for this disease in the 80s and in the early 90s, mitochondrial based drugs were quite popular, including coenzyme Q10 and creatine. These all showed really promising benefits in rodent models, and some even in non-human primate models, but these all went into phase 1 and phase 2 clinical trials without very much success in the clinic. And so while it is a unfortunate effect of mutant Huntington, along with lots of other things going on in the cell,
I think the focus now has been to try to lower expression of the disease-causing genes so that we can prevent all these downstream effects. But it's a great question.
Thank you. I have the last question. It's from Dr. Raymond Dudla to Dr. Yazdan Shemirad. Can the optogenetic approach be extended to deeper cortical areas, not directly under the dura? For example, areas within the posterior parietal cortex?
So we are getting expression. So our techniques helps us to get expression across all cortical layers. And depending on the opsin and the wavelength of light that you use, you can actually reach to deeper cortical layers. It depends on like how deep in the sulcus you want to get. So the evidence that we have right now is that about like 2 millimeters down to the cortex we can reach, but I don't know that we can reach beyond that. This is with either green or red light. So if I answered your question, sure.
Yes, thank you very much. In closing, I think this has been a wonderful session. I'd like to thank the speakers for their incredible talks and cutting edge work. And I'd also like to thank all the participants and everyone who has been on the call, and also everyone who is participating in the discussion. So thanks everyone for helping us celebrate this 60th anniversary. Thanks so much.